Hello, in this video we will discuss about the biogas production procedure and biochemistry. We will just understand about the biogas production first. So that is why the biogas produced as a methane gas which can be used in the burner for cooking or something or other things. So let's begin to understand from initial steps. So here is the herb, shrub and tree which that will be eaten by the herbivores. So herbivores will be eat, and after the eating the digestion will be occur, the cellulose digestion by cellulase enzyme and digest by bacteria, and assimilation that will be the part of the body, and egestion in the form of feces contain dung. Organic matter is known as dung, and water will be added. So let's begin to understand the gut microbiota in the ruminant animal. Contain Ruminococcus, methane producing bacteria, and bacteria work symbiosis as a type of mutualism to produce a short chain of fatty acid and vitamin Kl, and also cellulose break down partially digestion and go the glucose into simple glucose will be absorbed into the blood circulatory system. So that is a normal process. So let's begin to understand. We are using the waste or feces of the herbivores in the form of dung, and we will add the water, and we will add the cow and buffalo dung collectively, and here is known as the Suluri cattle dung and water will be added into the inlet mix tank. So this is a mix tank. After the mixing of these that will be mixed and add the water. So this is the ground level you can see, and this is I am drawing the bigger picture for understanding the biogas procedure and the biogas production procedure. So in this way, here is the inlet pipe. Inlet pipe will transmit the dung organic matter into the floating gas holder type biogas plant. This is, and we will add the into the underground digester tank. So this is the underground digestive tank which contain partition wall, and this is the anaerobic condition will be occur, and anaerobic bacteria will be act for further breakdown of the other things, and carbon dioxide will also use that bacteria, and bacteria use carbon dioxide to produce methane that is known as the methanogen, and it is used the energy from the decomposition of the dung. And after this, here is the biogas will be occur. After the pressure will be increased, so the biogas collector will be put in this way and set the wall, gas wall, and the biogas collector will be here. So in this way, here is the one hole will be created between the partition wall. So after the pressure will increase, so increase the dung, and in this way the movement of the movement into the partition wall, and here is a sport for pipe, and this is the outlet pipe, and in this way the creation of the pressure will be created due to the gas. So the gas create more pressure, and gas will move outlet of pipe, movement of the outward the spent slurry matter into the outflow tank. So this is the outflow tank, and this will be waste and can be used as a manure. Manure can be used for the plant growth and development, and plant is important for the manure due to the organic matter still here. And here is a floating gas holder, and you can see this is the floating gas will be give the pressure to move upward the dung in the form of spent slurry manure. So in this way, here you can see the outlet for biogas. We can set the valve for providing the gas to the burner or something. So let's begin to understand the methanogenesis. So the hydrogenotrophic bacteria, methylotrophic bacteria, and acetoclastic bacteria, which that is found the methanococcus M and methanococcus methanocarcina, and methanocyta. These all type of bacteria used for the methanogenesis. So let's begin to understand the biochemistry first of hydro. So the hydrogenotrophic, which the hydrogenotrophic bacteria, which that will be fix the hydrogen, and hydrogen come from the hydrogen which that is released from which that is synthesized from the bacteria, other bacteria. And here the methanofurane, which that is bind with the carbon dioxide, and after this binding the ferredoxine reduction phase to oxidize, and oxidize it is again the oxidize to reduce after the hydrogen fixation, and here is the formyl methanofurane which that is produced, and in this way the methanofurane will be fixed the tetrahydromethanopterine, which that will be released the methanofurane. And after the releasing of the methanofurane, the formyl tetrahydromethanopterine, tetrahydromethanopterine, which that is formed. And in this way, the hydrogen will be fixed in the presence of the formyl dehydrogenase enzyme, which that fix into the water, while the formyl will separate the hydrogen, and in this way the methynyl tetrahydromethanopterine formation will be occur, and in this way the F enzyme, this F coenzyme which that containing the reduced form to oxidize and donate the hydrogen, two hydrogen, and again the one hydrogen, and again the two hydrogen will be give through a hydrogen gas, and this hydrogen gas come from the bacteria reaction, and in this way here is the again reaction will be occur, and here is the MER is the enzyme used for it for the fixation of hydrogen, and to produce methylene tetrahydromethanopterine, and in this way the methylene tetrahydromethanopterine will be formed and containing a CH3 bond. So the whole formula I am not drawing here, and here is the HS coenzyme M, which that will be separate the tetrahydromethanopterine, and after this formation the methyl S coenzyme M. So the coenzyme M will be added while the tetrahydromethanopterine will be separate, and in this way the HS coenzyme B will be separate the coenzyme MS, and the S coenzyme succinyl coenzyme B. So this is the succinyl coenzyme, and in this way the coenzyme will be separated and again useful, and in this way you can see this is the methyl succinyl coenzyme M will be formed, and in this way the methyl coenzyme hydrogenase enzyme used to produce the methane, and in this way the methylotrophic bacteria use not use the hydrogen, but it will be give the reverse reaction, so it will produce the methyl succinyl coenzyme M. So the methyl succinyl coenzyme M will be produced through a methanol, monomethylamine, and methyl thiol, etc., will be stimulate and produce these things. And in this way here is, as you can see, this is the MCH will be formed. So remember about that. This is the basically the methyl succinyl coenzyme M will be methyl coenzyme H will be separate, and the methane formation will be occur because the methyl coenzyme which containing hydrogen, so the carbon fixation of the hydrogen will be occur, and in this way it can be reversible reaction because it can be reversible, and in this way here is the HS coenzyme M, the succinyl coenzyme M with the MTR in the presence of which that is the tetrahydromethanopterine succinyl pterine protein will be really bind to again that reaction will be reversed, and here is you can see this is the MER used to produce again the F coenzyme hydrogenation, and the MTD used for the methyl methylene tetrahydromethanopitarine dehydrogenase hormone, and in this way again the MCH is used for the methylene formyl methylene tetrahydromethanopitarine to form a formyl H4. So in this way, that is again the reversible reaction can be a reduction of the ferredoxine reduction, and oxidation again that is the opposite. And on the other hand, important thing is that here is the acetoclastic bacteria use the acetate production, and ATP used to produce the ADP and acetyl formation to become a succinyl coenzyme A will be separate the phosphate group, but add the acetyl CoA formation to separate the carbon dioxide to, and the carbonic anhydrase enzyme to produce carbonic acid, and the coenzyme dehydrogenase enzyme used to produce again the methyl tetrahydromethanopitarine for production of the methane.